Morning. Folks are still entering, so we'll give it a few minutes. Thanks for joining us today, everyone. All right. Looks like attendance is leveling off a bit, so I will get started. Good morning, everyone. My name is Leora Fox. I am the Assistant Director of Research and Patient Engagement at HDSA. And I am joined today by my colleague, Kelly Andrew, who is the Coordinator of Research and Mission Programs. Want to say hello, Kelly?
Hi, yes, good morning, everyone. Excited to be here.
Great. So today we're going to be hearing from two speakers who are both HDSA supported fellows through the Human Biology Project. And before I introduce them, I want to just go over a couple reminders about how you can interact with us and our speakers during today's webinar. And I'm going to point you to a couple of additional HDSA research resources and upcoming webinars. So we're going to have two presenters today, so we'll do a Q&A after each one. But at any time, you can send a question through the chat function in your toolbar. Only HDSA and the presenters are going to be able to see your questions, not the rest of the audience. But at the end of each talk, Kelly and I are going to help to field those questions, and we'll do our best to follow up on any that we don't get to. Just a reminder that we record all of our research webinars and make them available on our YouTube channel and our webpage within usually a few days. So if you want to watch anything you've missed or share this with somebody else, you will absolutely be able to do that. And if you are new to HDSA's research resources and are looking for ways to learn more about HD clinical trials and participate, I recommend checking out hdtrialfinder.org. That's HDSA's clinical trials matching service. It is a website and a call center where you can read about and learn about ongoing recruiting trials in the US and Canada, or you can anonymously enter some very basic medical info for yourself or someone you know and get matched to a clinical trial, and even notified of trials that fit your profile in the future. We also recommend checking out hdbuzz.net and hdsa.org/blog for HD research news in plain language. Enroll HD is a great way for anybody in an HD family, no matter what your relationship is to HD, to get involved in HD research. We have some more HDSA webinars coming up this fall in our HDSA and Me and Disability and Research series. So on October 7th, we have an HDSA and Me panel discussion on managing behavior in HD. November 3rd, Jane Paulson and her team at Wisconsin are going to talk about the Prevent HD study, which is an extension of Predict HD. And this is an observational trial that's helping us to prepare for prevention of HD. November 9th, there's a disability webinar upcoming with Allison Bartlett, our resident disability attorney. And on December 7th, we'll have an Ask the Scientist Anything panel with all of the editors of HD Buzz. And without further ado, I'm going to introduce our speakers. So I'll introduce both of them, and what we're going to be starting with Dr. Simon Lagonière, who is an instructor in neurology at Harvard Medical School. He sees HD patients with neurology and neuropsychiatry needs at the HDSA Center of Excellence at the Beth Israel Deaconess Medical Center in Boston, Massachusetts. And his research interests include neuroimaging and techniques for altering brain activity, such as transcranial magnetic stimulation. And his work has focused on understanding how networks of brain cells communicate in health and disease, with a particular interest in executive functioning. And in addition to his research and clinical work, Simon has worked on several treatment trials in both Alzheimer's disease and Huntington's disease. And he was a recipient of the 2020 HDSA Human Biology Project Fellowship to study neuroimaging biomarkers in people with Premanifest HD. And his project aims to better understand disease progression by capturing early changes in brain networks using a longitudinal study design and a novel approach to assessing functional networks. I am also going to introduce Dr. Michael Plazek. who is an assistant professor in radiology at Harvard MGH. Michael's the director of pharmacology at Martino Center for Biomedical Imaging, and he runs the chemical neuroscience program, where he focuses on the process of moving chemical discoveries towards real new methods for doing brain imaging in humans. And a major area of study for his lab is how the immune system contributes to brain disease and developing ways to visualize early disease changes in the living brain. Michael was a 2018 HDSA Human Biology Fellow, and during his fellowship, he worked on a new way to track brain inflammation in HD. And as a direct result of this project's success, his team is preparing now for first-in-human studies of a radio tracer, and their hope is to visualize how the immune system might be disrupted in the HD brain to enable development of early intervention therapeutics. So I'm going to hand it now to Simon. Thanks both for being here, and we're looking forward to hearing about your research.
All right, thank you very much. Let's see if I can share my screen. Leora, do you guys see the?
No, we're not seeing it.
Yeah.
Are you able to select? Let's see here. Let me try that. Give me 1 second. Yeah.
We're just seeing your faces so far.
All right, this may be a little better. Does that work?
That's looking good.
All right, we made it.
Great.
So thanks everybody for joining. Thank you to the HDSA for providing some funding for this project and these ideas, and also the platform to share a lot of this information with all of you. And I also want to thank all the patients and families that participate in a lot of these human subject research endeavors and trials, because obviously without you, this information would remain hidden away. So starting with that, as way of a little bit background here, I have no conflicts to report. So I'm a cognitive neurologist, and I really spent the last decade or so of my life really focused on cognitive disorders of all kinds in the neurological realm. And I see patients, the majority of the time I've seen patients with myriad of different forms of dementia, Alzheimer's disease being the most common. But across the board, that's been really my interest. Over the last few years, I've spent and divided my time a little bit with a different patient population, including patients with Huntington's disease. And I work both in the cognitive neurology division at Beth Israel in Boston, as well as as we mentioned, the Center of Excellence here at Beth Israel. And I work closely with Dr. Sam Frank, who's been really an amazing mentor and taught me a lot about Huntington's disease. And we have a number of different individuals in our team who really help out, and it's a really a great team to be part of, to be honest, and a really great community. And I've learned a few things over the last few years, working directly in clinical trials, but also in the outpatient world with patients with Huntington's disease and their families. And the first thing I've really that came to me after working a couple of years was that this is really an exciting time in many ways to be working in this field. And one of the reasons I transitioned over, so for a number of reasons, Huntington's disease, as opposed to other types of dementia, is really a model system for understanding neuropathology and the progression of potential underlying conditions over time. There's also a lot of, in the pipeline, there are a number of different therapies that are being advanced in different, attacking the problem from different angles. And because of that, I think it's just really interesting to be a clinician scientist and trying to sort of position yourself right, at key moments in the development of certain treatments. And that, I think our understanding of these types of pathologies really expand out, maybe in the hopes of helping other forms of neurodegenerative diseases. I'd say that, the things that may be sort of obvious to everyone on the call is that, in many ways, Huntington's disease is particularly, in both ways, it can be a burden to families and patients because of this knowledge that you have about the genetic transmission and this long prodrome where you're in potentially a pre-manifest period, and then you slowly move into another clinical category. And it just really becomes a real challenge to figure out, I think, what symptoms are part of one category or the other, or when is it that symptoms have progressed to a point where we can, our diagnosis changes. And I think this is really a challenge for a number of different reasons, but it's also scientifically, I think, a really interesting opportunity to kind of look in and see what we can do about figuring out this trajectory, and if there's anything we can do to alter it. And so this is not a new concept, of course. This is taken from Scale et al., just a really interesting paper last year, looking at early biomarkers, molecular biomarkers. But the general principle, illustrated here is just there's really a decline. It can be a decline over many, many years in many different realms, including a cognitive realm that interests me in particular. But these are over many years, and it's finding out when a diagnosis should be made or when an intervention should be timed has become a critical question, I think, especially in light of a lot of promise in terms of potential therapies. So biomarkers being a real important thing to go after. The clinical tryout is really, when I learned it in medical school and really before seeing too many patients, it's really described, as a triad you may be familiar with, but really it's predominantly classified as a mood disorder, with cognition being subsidiary or really sort of thought of potentially as an add-on later on. And the diagnosis is really, clinically is really defined most by the movement portion, the motor domain, deficits in the motor domain. But being mostly focused on cognition, I kind of think of it in this way, where cognition takes the forefront. And that's sort of germane to a lot of what I do. But there are many reasons for this, and the focus on cognitive changes is motivated by a number of different reasons, a number of different forces here. So cognitive changes can be early, and they can predate even the motor symptoms in many cases by years or even decades. We don't really have a clear treatment for the cognitive portion. There's no directed therapy per se yet that's substantial in altering some of the symptoms, some of the cognitive issues that may arise. And there's really, in my view, an outsized impact on patients and families when you think about even a small change in cognition or cognitive abilities. Really means a lot, really has a big impact on daily life, whereas a slight change in chorea may not have as large an impact. It could, but in general, and in many cases, it doesn't. And a lot of our sort of daily life and functioning and professional lives are dependent on our cognitive functioning, and a small change there can really mean a lot. So, with that in mind, I conceptualize this probably pretty simplistically, looking at this progression over here, starting on the left. There's a molecular pathology that's driven really by the mutant Huntington protein. There's misfolding that occurs. There's an eventual progression to a point where over here, where that cell itself is having difficulty. So there may be interruptions in vesicle trafficking or even in communication between cells. At some point, that has a larger effect. And we're not just talking about cells anymore, but groups of cells, and maybe some regional effects where cells are talking to other areas. Neurons are talking to other neurons in the brain. And we're really talking about something large, or something like brain networks in particular. When those networks break down, at some point, well, I should say rather that at some point the cell loss, so the neuronal death and potential loss, in particular in the striatum, but also cortically and outside of the brain, really leads to structural changes, and those have been observed for some time, and those are potentially progressive over time and may predate some symptoms. And finally, that culminates over on the right here with overt cognitive symptoms. So particular early changes in executive functioning, some processing speed issues that have effects across the board, both in terms of cognition, but then in terms of daily life. And these are really important deficits that it may be challenging to retrain or to provide supports for. So that's the general way I think about it. And really the way that I'm thinking about intervening, or the thoughts that come up in my head are, well, what is sort of the right time to figure out the most important change? And for me, this regional connectivity, the communication between neurons at a larger scale is really something that's interesting to me and something I've worked in or with for some time. But it's also the point at which I think communication starts to break down across the brain. And possibly later than that, there's been, a substantial amount of disruption or even destruction. And prior to that, there may be cellular-based changes, but it's not clear whether these are having a clinical impact. So that's kind of like the sweet spot that I think about in terms of what I'd want to go after if I could, in terms of the progression of pathology. So of course, I'm not the only person to have done this or think about this. In the last many years, there have been attempts at categorizing changes, neuroimaging changes and biomarkers in both Huntington's disease and the pre-manifest period in particular, as symptoms progress. And it really falls into two big categories in the MRI world. Well, I should say three, but in the functional MRI world, there's fMRI, where you're looking at really task-based activation. So we're looking at areas that are activated when a particular task is, or particularly brain areas involved in a task. And then more recently, really looking at functional connectivity MRI, which is really a means of dissecting the brain and looking at areas that connected to each other rather than looking at single areas that are activated with a task. And this is the one I'm going to focus on. So this functional connectivity MRI, here's a simplistic sort of representation. Really what you're trying to look at is, in the simplest of ways, is patterns of activity. So if you see these different areas that are highlighted here, and you look at this time course on the right here, what you see is this area over time activates and deactivates as it's processing information, presumably, and talking to other areas of the brain. And then you have the green area here that has a different time course. So it's following a different pattern, and it may be involved in some other function, or it may be in time, maybe sort of delayed, or it may be linked in other ways, and it may be competing for resources. And these are all sort of open questions, but these are time courses that you can derive. So activity levels can be monitored. Most of the connectivity to date has really been done in something called a resting state. So we place a participant in a scanner and have their eyes open for a while and ask them to think about whatever comes to mind over a few minutes. So they rest there comfortably, try not to sleep, and think about whatever comes to mind. And this has allowed sort of simplified protocols, really, because you can ask participants to do this and across different scanners and different locations. There's no particular task involved. So it's, this is really thought to reveal the intrinsic sort of connections between areas. What you end up with is a series of brain networks. So this has been known for some time, but even at rest, when you're just sort of laying there in the scanner with your eyes open, some areas, and these are represented in colors here, just at the a basic level, in a sort of simple, sort of simplistic way, you can think of the brain as divided into these functionally connected areas. So a lot of the areas in blue, for example, are mostly coinciding in time and activation patterns with other places that are blue. And this is thought to constitute a functional network per se. And you can divide the brain into as many networks as you want, but there are certain general principles here in terms of what areas are talking to what other areas. And I should say that, as I mentioned before, these are derived traditionally when someone is resting, so not engaged really in any task per se. And the idea has been to track some of these network rather than regions that are particular regions that are activated by a task. Can we look at whole brain networks? Can we look at areas that are sort of modulated and forming probably the underlying scaffold of the brain as it's processing information? And do these look different? Are these changes reflected in a population that may have a pre-manifest HD, for example? And so that's been sort of an open question. And there have been a number of studies looking at this. And I think the conclusions are not clear yet. I think there's part of the issue is that there may be many different ways of analyzing information, many different ways of putting this all together and coming to certain conclusions. But the reality is that this data is inherently noisy. So having someone in a scanner with their eyes open, the acquired data is not really, you see those time courses over time, but it's just inherently noisy. The other potential issue with it is that it's unconstrained. So we don't, other than giving general instructions, we don't have individuals doing necessarily the same thing. And the reproducibility is an issue because, as you can imagine, you may not be resting the same way on any two occasions, even within the same day. And individuals may rest very differently. So you may be an individual who's sitting in the scanner worried about something that happened yesterday to your family, and someone else may be thinking about, maybe their shopping list for the rest of the day. And someone may be really anxious about being in a scanner. So you can imagine the brain may be configured in a slightly different way. And there's really an argument for saying that as you're engaged, when you go from a resting state to a task state, you're really potentially engaging different areas, and the areas are dynamically remodeling. Certain areas start talking to other areas. So what you get when you're doing a task is really actually a different set of areas that may be connected to each other rather than at rest. So how should these data be acquired? And that's really some of the question I think is sort of open, and one that I'm interested in. And what about looking at brain networks during a task? So can we capture some of this in a way that's a little bit more constrained? Can we look at functional connectivity and brain networks when they're in a different state, a state that's possibly stressing them out a little bit, potentially during a task that really pulls on those different domains that we think may be affected early in pre-manifest HD. The other benefit of looking at this is that in this way with a task is that every participant's really doing the same task. So we kind of force them into a certain paradigm, and that helps with the statistics later when we know that we can kind of confine these things. And it's probably a little bit less noisy that way. And if we were going to design a task, what would it be? And ideally, you'd want to get something that is hard enough so it engages everyone in a similar way, but not too burdensome or difficult that participants either give up or kind of are not able to keep up. This has been, so I chose this task, this task I've had some experience with. It's a pretty easy task. It's called a continuous performance task. And really what you're looking at on the left is a picture of a city, on the right is a picture of a mountain. And these were slowly morphed one into the other. Your task is pretty simple. You just press a button if you see a city, and don't press if you see a mountain. And these images are flashing at about 1 a second. And they're kind of going from one into the other. So you do this sort of boring, more or less boring task for 10 minutes in a scanner, and you're pressing the button, and you do the best you can throughout the entire scan. And this provides a platform for really looking at brain networks during a similar period rather than the fluctuations that may occur when you're simply just resting. There's some evidence for this that there may be the task state may be a little bit more constrained and a little bit easier to look at in terms of managing data. So this is for my own work, and it may not be obvious from these graphs, but on the right here, when you move from really a rest state to a task state, what you see is that networks that were more or less aligned at rest are now kind of moving in a very structured way during that transition. So when they're engaged, when you look at these networks in an engaged state, they actually look different than when they're in a task state. And we can actually predict performance on that task in the scanner quite a bit better, just looking at two networks in particular. This isn't an optimized study or anything. It's just looking at two very well-known networks that are kind of mirroring each other and working against each other potentially. And looking at those two, we can really predict how well you'll do on this task for over 10 minutes. And that was a project I did in healthy controls. So there's some evidence that this might be helpful. So really what it comes down to is really, for the patients with HD in the pre-manifest period, do their brain networks look similar to other controls that we've seen before at rest, or do they reorganize differently? Is there a signal there that we can see? And so the approach is really to use that task that engages their attention to some degree, a little bit of processing speed, and an element of inhibition. So when you're pressing the button to see the mountains or the city, you have to withhold and prevent yourself from pressing if it's the incorrect image. Then, as I mentioned before, it's really a constrained task rather than resting state. And if none of what I said sort of made sense at all, or if it was simplistically boring, or if it was contrived and complicated, this may summarize a little bit of some of the thoughts here. Really straightforwardly, looking at this red line versus a green line, we're moving this whole line downwards and trying to pick up a signal earlier on in the pre-manifest period if we can, to optimize what we talked about earlier, is really monitoring and detection and potentially seeing changes. So you imagine if you're looking at 2 time points in a longitudinal study up here, depending on what modality you're looking at, you may not see a change. With others, the slope here is different. You may see a change. And this may detect something a little bit earlier and signal that something is happening now that wasn't happening before. So the way we designed this was really to recruit from our clinic and select 20 patients that had we considered to be in a pre-manifest period, and match them in terms of age, sex, and education with controls. They came in. We characterized them along neuropsychological realms on the task listed there. We also made sure that these individuals, really we thought clinically were in the pre-manifest period. So those are the ones we wanted to look at. Then we went ahead and obtained structural data and the fMRI that I talked about, both at rest and during this particular task. And that was it. That was sort of a pretty simplistic design. And the idea is to look at this again. So once over the last few months during the beginning stage of this project, and then again at 6 months and 12 months, with the idea that maybe we can capture some longitudinal change and compare that to changes that we know are potentially happening. So other biomarkers, whether they be neuropsychological tests or measures of structural brain changes. And so this is very early on, and we have recruited probably between 50 and 75% of our goal. We're getting there. But I wanted to share some initial results that I think I find interesting. I hope you do too. And these are sort of along 2 different realms. So this is looking really at those 2 networks I talked about. This is really the highest level analysis, really the first analysis. And looking at, in blue are patients with pre-manifest HD, and in orange are healthy controls. And what we see here is a little bit of a difference here in the default network. This has been reported previously, but it's really, this is within the network itself. So these areas here are talking to each other during a task. We don't see much, actually, interestingly, in this network here, the dorsal attention we thought may be engaged in this task. Doesn't seem to be a big difference there, but the difference seems to come out of the default network during the task itself. And over here is really an anti-correlation between these two networks. And that's probably primarily driven by this, the change in the default network. And that may not be something you see at rest. So that was potentially brought out by the task. And maybe to drive that home a little more, here was another high-level study we did here looking at our two different groups. And this, again, was at rest. And over here was during the task itself. And if you can follow this, this was the scan. This is mapped out onto the surface of the brain. This is really a functional connectivity analysis using the putamen part of the striatum, using that structure in particular, and looking at seeing what this was connected to in our different individuals. So at rest, we see our patients with Premanifest HD have this pattern. The controls have this pattern. And then when we do subtraction over here, on the left still, we see this pattern here. So sort of speckled a little bit all over the place. When we do the task, our pre-manifest patients actually don't do the task any differently. So their performance is similar to our controls, their age match and education match controls. But what we do see is that, I think, is that their networks look a little bit different. So during the task itself, the controls are engaging more areas or connecting to the putamen, really. And when you do a subtraction, what you see is a more convincing difference here between the two. So if you look at the whole resting state over here, the difference between our groups is small. And you look at here during the task, the difference is probably larger. And this is sort of, this could be, you could interpret this as that we're bringing out these changes, that this is an adequate cognitive stress test of sorts and bringing out a difference between the group that otherwise would not have existed over on the resting state side, and may be one of the reasons it's harder to pick up both cross-sectionally at one time point and then throughout time as longitudinal studies have tried to pick up these differences. So the initial basic prelim results look pretty encouraging. These were some methods, the fMRI, if anyone's interested. So the future directions, there's many ways in which you can take this, but I think the initial obvious ones are looking at putaminal connectivity. So the putamen, we know, is one of the structures that really seems to be affected early. So what it's doing and what it's talking to across the brain is probably really important to know. And that was the study, the initial study we did. But looking at this in more depth, breaking the putamen into segmented parts may be of interest. And then whole other brain networks as well, because this is pretty exploratory. If this is indeed picking up something new and novel, there are a number of different ways to take this. Then of course we'll do a 6 to 12 month follow-up. So that was the project, and that's how we're going about it and the initial results. I want to thank again everyone who's participated in this study. I'm always really amazed and inspired by the patients who are giving up their time and energy and coming here and participating in all these studies and trials. It's really quite amazing. The HDSA for this platform and providing some of the funding, and then the team at Beth Israel, including Simfree. And so that's it. So happy to take any questions.
Thanks so much, Simon. It's really nice to see some of this usually very complex imaging data broken down in a way that can be understood. I'm going to give, we have time for a couple of questions. I'm going to give folks a minute to type anything into the chat that they'd like to, but I can get us started. I am wondering sort of what is your ultimate goal for this type of imaging? Like, could you see it being an outcome for a future drug intervention trial, or what do you think is the
yeah, I think the next steps are really validating this, because this obviously is a relatively small cohort, so we need to make sure this is a true finding, really. I think the idea is really to potentially incorporate it as a biomarker of sorts, a neuroimaging biomarker. I think that there is, whenever you have a task set up, it's a little bit more difficult in terms of a protocol than having someone rest in a scanner. So there are logistic challenges to developing this at a larger scale, but something like this, or a task similar to this that engages networks, I think may prove to be a little bit easier than just a resting state per se to see. So I really hope that that becomes a marker that's useful. It probably will be in combination. It probably won't be alone. It would be in combination with some other imaging and other markers. So I think probably, you know, there are predictive formulas out there. They're not absolutely perfect, but they provide a lot of the variance in information. This may add on top of that. So you might think of, you know, CAG repeat plus, you know, a putaminal or striatal structural atrophy plus a functional connectivity component on top to make as best a prediction as possible. I think these really fit into prediction models more than you know, more than anything else.
Thank you.
And you really need, I think. Oh, sorry.
No, go ahead.
No, I think eventually, you know, I think these are powerful techniques longitudinally is the other thing. Like you need to really look at two sort of time points for a lot of this imaging data, because a single one is just difficult. Everyone's brain's a little different, and these techniques really work when you're your own control over time. So, you know, I think having a couple scans might provide you with, you know, a change there that will be predictive.
Thanks. I'm seeing one question in the chat. I want to respect everybody's time, but so we'll just do this. Well, very quickly, someone's asking what power was the MRI, 3 or 7?
Oh, it was 3.
And another question that I like here is whether you used humor as a part of your study. Was that kind of emotion incorporated in there, or is it really just purely visual?
Oh, you mean in the task itself, probably,
yes,
presumably.
I think so.
Yeah, it was unfortunately. It's an interesting question, because activating emotions is another area of interest, obviously. But this was a pretty boring task. Is a grayscale city or city or mountain, and unless that excites you for any reason, one way or another, it was not meant to be an emotional task.
All right. Well, I think in the interest of time, we're going to move on to Dr. Michael Plazek. But thank you so much, Dr. Laganière, for this presentation. And if any other questions come in, then we'll do some follow-up. Michael, are you able to share your screen?
Yep.
There you go.
Does that look okay on your end?
Does indeed.
Okay. Thank you, everyone, for attending today. It's a really great honor to speak to all of you and share some of the work that has been supported by HDSA. And thank you for the introductions from Leora. This is a project that has been ongoing for about 6 years now, dating back to about 2015. And it's a large collaboration by a few different teams. The imaging team being myself and Jacob Hooker at the Martino Center at Massachusetts General Hospital, and then a medicinal chemistry team at the Broad Institute led by Dr. Flo Wagner, and our Microglia biology experts at Children's Hospital and Harvard Medical School, led by Beth Stevens, and also a lot of the work from the Stevens lab was done by a prior HDSA human biology fellow, Dan Wilton. So, like with most problems and projects at the very early start, we try to lay out, in very simple terms, the problem we're trying to address. So here, we're trying to develop highly effective, disease-modifying therapeutics for Huntington's disease, and we need to understand neurobiological changes that occur at the earliest evidence of disease conversion. We need to alter this potential damaging biology prior to irreversible damage, such as tissue loss or atrophy. And we need to be able to stratify novel therapeutics with tools that can really inform us if a damaging biological process is being restored or healed prior to waiting for symptom improvement. And critical to this mission is identifying the right tools that can do just this. So sort of the approach that we're taking solving this problem is really the only way to study neurobiology at early disease conversion in the living human brain is with a non-invasive imaging technique such as MRI or positron emission tomography. So where do we start? So to start, we typically ask ourselves a series of questions. And the first question being, of all the regions of the brain, which regions show early evidence of disease conversion and are related to HD pathology? And question two, of all the cells in that brain region composed of the basal ganglia, which is composed of the globus pallidus, caudate, putamen, which cells in those brain regions demonstrate unhealthy function immediately upon disease conversion? And of all the proteins expressed by those cells, of which there are thousands, which proteins demonstrate significant changes in these cells in the basal ganglia at early disease conversion? So the technique that we're using to sort of address these questions and problems is a molecular imaging technique called positron emission tomography. And really it stems from, and the reason why we need this technique is stems from our understanding of and really ability to treat all CNS diseases is really profoundly limited by our inability to access living human brain tissue. And neuroimaging with a technique like PET allows us to see into the living brain by providing molecular signatures of biochemical processes. And that's where my research focuses on, is using this particular type of imaging device called positron emission tomography to visualize changes in brain biology of the living brain. PET is a type of imaging modality that uses essentially a camera and it's a very expensive camera that's capable of measuring in a quantitative way concentrations of molecules in the living human brain. So what do I mean by positron emission? This is a certain type of radioactive phenomenon where an unstable nucleus, which we make, expels positrons until this nucleus is stable again. And these positrons send off light. This light is called photons. And this light is picked up by our cameras or our PET detector. And after millions and millions of these collections are performed, and this happens continuously until these molecules decay, we can generate pictures or tomograms. And there are other types of tomographs that you may have heard of. CT is X-ray computed tomography. MRI is magnetic resonance tomography. But in the case of PET, it provides pictures of molecules, and this imaging is termed molecular imaging. So PET is a type of molecular imaging. So you might be asking, why are we imaging molecules? I thought we were imaging the brain. Well, the brain is full of molecules, peptides, proteins, small molecules, and these all carry out a variety of functions or neurochemistry. And if we want to image a particular function of the brain, well, then we choose the corresponding molecule that signals for that function. So PET requires the use of very, very special molecules, and these molecules emit an energy that is picked up by a PET detector. And after millions and millions of these collections, we can start to generate 3D images of concentrations of these molecules in the tissue we're interested in. In this case, it's the brain. So this slide is sort of working backwards from imaging back towards the development of these radio tracers, which all occur on the day of an imaging scan. So these molecules are identified and tagged with a small energy emitting isotope. And this isotope gives off a very specific energy that is picked up by our PET detectors. We choose a molecule that targets a particular protein that we're interested in studying, in hopes that this will be a biomarker to stratify, for instance, efficacy of a novel therapeutic. This entire process of developing a radiotracer occurs on the day of the imaging experiment. Because of these high energy isotopes decay so quickly, this whole process is sort of done in a very expedited but safe manner to develop a sterile formulation that's safe to be administered to patients, and then can eventually inform us on the targets we're interested in studying. So here's an example of a PET imaging study. This particular imaging study was conducted by colleagues of mine, and actually one of the principal investigators on my project was the lead principal investigator on this project, Dr. Jacob Hooker. So this was an imaging study that was done at the Martino Center, published in ACS Chemical Neuroscience in 2018, that showed that a particular protein called the translocator protein, or TSPO for short, and what the investigators wanted to study was sort of the neuroinflammation signature that TSPO can provide in Huntington's disease as a way to determine and start to understand the component of the neuroimmune system in neurodegenerative diseases, and in this case, in Huntington's disease. So in this case, in this slide you see here, there are 8 images of different subjects that underwent a PET scan with a particular molecule called PBR28, which targets a protein in the brain called TSPO. And then it essentially provides a concentration map of low expression to high expression, low being black or blue colors and high expression being red or white, of this protein in certain regions of the brain. And in this particular slide, there are 7 HD patients and 1 healthy control. And if you look at these images long enough, you can see that there are certain brain region, specifically the caudate putamen, that has a lower amount of this protein called TSPO in that brain region. And this was really one of the first studies that showed us the neuroimmune component is relevant to HD pathology. One of the challenges with TSPO imaging is, while it's helpful at informing us that there is a general neuroinflammatory signature, it's not specific to a certain cell type that we think is most important in studying the neuroimmune system, and that's microglial cells. And unfortunately, we haven't been able to mechanistically link TSPO upregulation to disease pathology. So I mentioned microglia. So microglia are the resident immune cells in the brain, and their primary job is to maintain brain health or brain homeostasis. And they are excellent at it. They have the ability to survey their surrounding environments with these long branched extensions of their cell body, which kind of look like tree branches without leaves. And they are able to detect any abnormalities nearby, such as other damaged cells. And they can try to clear these problems before significant damage can occur. And actually one of the functions they can perform is they can actually eat other dead or dying cells. And this is termed engulfment. So microglia, perform many important functions, and their activity has been linked with a number of major human brain diseases, including different forms of neurodegeneration, including Huntington's disease. So microglia are able to help the brain maintain a healthy brain environment as a key component of the neuroimmune system. And when there's small local damage, such as the middle picture here in this slide, microglia have sensors that they can alert if there's a problem, and they can migrate towards that area and help restore function or brain health. So they're able to repair small acute damage. When there's a larger insult or a stronger insult to the brain, such as an infectious challenge or significant tissue injury, this may trigger more drastic changes in the way these microglia behave. and it may lead to substantial impairment of neurons and other cells in the brain. So my research aims at understanding this switch point when microglia transform from helper cells to damaging cells. And there is significant evidence now that in neurodegenerative disease, including Huntington's disease, that this occurs very early in disease conversion. So we feel that microglia-based diagnostic approaches, such as neuroimaging with PET, with chemical probes or molecules that can target these microglia and target key signaling pathways that microglia are involved in, may allow for the detection of at-risk individuals much earlier than is possible with biomarkers directed at other cells in the brain, such as neurons. And then we also feel, probably more importantly, is that microglia-based biomarkers will permit the monitoring of disease progression and recovery of novel disease-modifying therapeutics prior to the alleviation of symptoms. So we know that microglia are important at regulating the neuroimmune system and have been associated with dysfunction in neurodegenerative disease pathology. So we took a look at some of the key proteins that are expressed by microglia and what their function is. And we used a particular type of biochemistry experiment that measures protein levels extracted from tissue to try and see if a certain enzyme called cyclooxygenase 2, or COX-2, is differentially expressed in brain tissue regions in HD patients relative to healthy controls. And excitingly, what we saw is that in blue here on the graph on the right, let me try and see if I can use the laser pointer. Graph here on the right, you can see that the COX-2 enzyme is significantly upregulated, almost tenfold, in HD brain tissue relative to healthy controls. And this dramatic increase in expression is only occurs in microglia cells relative to other cells that are in the brain, such as astrocytes and neurons. So this is a very exciting finding. We then extended this work out to measure protein levels in manifest and pre-symptomatic HD tissue, and saw a significant increase in COX-2 levels, almost tenfold compared to healthy controls, while other markers of microglia activity, such as TSPO, which is the protein that I showed the imaging data on before, does not show significantly altered levels in early HD or in pre-symptomatic HD. So just sort of the take-home message here, without getting too detailed on all the biochemistry, is that basically our data shows that COX-2 could be a superior biomarker of microglia activation. And this seems to occur early in disease conversion and prior to symptom onset. So we have our target, we have the enzyme that we're interested in studying, we know the cells that we want to study, that's microglia, and we know the enzyme we're interested in studying, that's COX-2. How do we develop a imaging agent or a molecule that can target COX-2 in the brain and help us study the concentration of that enzyme in the living brain? So it starts with a chemistry approach. So we, through over about 5 years of chemistry development, we have an excellent collaborative team at the Broad Institute who synthesized more than 150 molecules that are specific for COX-2. And we assess them for a number of pharmacological properties that are very important for developing a positron emission tomography radiotracer for brain imaging. And we sort of selected our best-in-class molecule after sort of an array of testing, pharmacological testing. And we started to tag each one of the best-in-class molecules with a small isotopic labeling tag, which will emit now an energy that our PET detectors will be able to pick up. And we started to assess these molecules in vivo. So the first translational step in developing a human brain PET radiotracer is to assess them with preclinical imaging, first in rodents. And we were pleased to see that our lead molecule, BRD, which we call BRD 1158, exhibits extremely high brain uptake in vivo in rodents. This is the first hurdle that we have to pass with developing any brain radiotracer for PET imaging. And then it's sort of a next experiment is to say, okay, it crosses the blood-brain barrier, which most molecules do not. Is it specific for the enzyme we're interested in studying, which is called COX-2, in the living brain? And this experiment here uses a very particular animal model that overexpresses COX-2 in one particular brain region, highlighted by the, identified by the crosshairs you can see here. And what this data shows is that this molecule very specifically targets COX-2, areas of the brain that have significant COX-2 overexpression, with red being high areas of concentration and black and blue being low areas of concentration. So the first two hurdles have been met. The molecule crosses the blood-brain barrier, and it's highly specific for COX-2. The next step is to assess these same parameters in a species that is similar to humans, in non-human primates, and baboons. And we see that these, this radiotracer has, hold on one second, my daughter just came in. Okay, we're good. This molecule exhibits high brain uptake in non-human primates as well. So this is sort of our last hurdle in sort of the translational pipeline of developing a human brain PET radiotracer. So where are we now? So our COX-2 targeting PET radiotracer has demonstrated excellent pharmacological and in vivo properties warranting translation to human imaging. So after around 5 years of development, we're now at preparing for an IND application for first in human PET imaging, which we are concluding the IRB and IND applications now. We hope to be imaging first in human in early 2022, hopefully by February is our target month. And then we'll soon be recruiting HD patients for research studies to examine COX-2 if COX-2 levels are significantly upregulated, and this will be shown by an increase in BRD 1158 binding in those brain regions in the globus pallidus relative to healthy controls. And the plan is to start with a small subject population, and then hopefully if there is early evidence to show that COX-2 is upregulated in pathological regions, that we will extend this study to a much larger group and potentially study them longitudinally. Oh, sorry, click twice. There we go. So first, I just want to thank first the Huntington's Disease Society of America for supporting a significant portion of this work, and really their support came at a really crucial time in the project where we had developed a number of molecules that we were excited about, but had not yet tested them in vivo with PET. And the support from HDSA allowed us to translate them into preclinical imaging and sort of assess their in vivo properties with hopes that we would be able to identify a best-in-class molecule that, and we did, and translate that into humans, which we hope to do in early 2022. So this work was done by a large collaborative team spanning across MGH, Boston Children's Hospital, and the Broad Institute. So I want to thank all of the co-investigators, all of the research team that helped carry out a number of these studies. And then also want to acknowledge the Martino Center Radiopharmacy and Pet Center for their help with this project, and the MGH Huntington's Disease Center of Excellence. So thank you, and I'll take any questions that we have.
Thanks so much, Dr. Plazek. It's really exciting to hear that this project is moving forward and has resulted in an IND. I will ask you to get us started. I know we're at the hour, so I want to respect everybody's time, but I'll ask you a similar question that I asked Dr. Laganiere, which is sort of, where do you see this going? Is this the kind of thing that, one day somebody's going to walk into their neurologist's office and the neurologist is going to say, okay, I'm going to give you a COX-2 scan, or is it more kind of research based as therapies are developed in the realm of immune system treatments for HD? If you could expand a little
Sure.
on that, that'd be great.
Yeah, and I think in the near term, it will be to stratify novel neuroimmune modifying treatments. It's challenging when we don't have these imaging agents or biomarkers that can really tell us mechanistically if we're modifying disease sort of at the biochemical level. And it's very difficult to do in living subjects. So techniques like imaging help us understand if the underlying biology is being improved in a way that will eventually lead to improvement of symptoms and hopefully slowing or halting disease progression.
Thank you. I'm not seeing a lot of questions coming into the chat right now, but we can certainly follow up if folks have questions. I just want to add that both of these investigators, as I know that Dr. Laganisiere's study is listed right now in HD Trial Finder and is continuing to recruit, and certainly we will be in touch as your study develops, Dr. Plazek. And we will list those as they are.
That would be great.
If folks are in the Boston area and are interested in participating in this important work. So finally, just to wrap up, I want to thank everybody. I want to thank our speakers once again. And thanks to all the participants for joining us today. And we'll see you in a few weeks with our next HDSA webinars. Have a great day, everybody.
Thank you.
Thanks, everyone.